One, General Biochemistry. We previously finished with coenzymes and cofactors. We looked at the structure of coenzymes, their sources, mostly from vitamin Bs. And we even looked at mechanisms of action of some of these coenzymes. We'll continue today to first of all look at factors that influence enzyme activity, and then we'll finish with mechanisms of action of enzymes. All right, so let's dive in. So we'll be looking at four major factors that affect enzyme activity. That is enzyme concentration, substrate concentration, temperature, and pH level of the environment in which the enzyme is acting. So we'll take each of these factors and look at how they influence enzyme activity. So the first, that's enzyme concentration. And we see enzyme concentration is represented on the horizontal axis, the rate of the reaction that the enzyme is catalyzing is on the vertical axis. And if you take a look at the relationship between concentration of enzyme and the rate of reaction, we see the rate of reaction increases with increasing enzyme concentration. Because this is understandable. If you have, let's say, if you have all things being equal, like you have concentration of substrates being constant, you have temperature being constant, you have pH also being constant, and you are only varying enzyme concentration. As you increase the amount of enzyme, you are increasing the number of molecules of enzymes that will be present in the reaction mixture. And so you have more enzymes that will be acting on substrate molecules. So in that case, you'll be having more products that will be formed faster once you increase the amount of enzyme in a reaction mixture. That is in an enzymatic reaction. So the rate of an enzymatic reaction increases with increasing concentration of enzymes. Okay, so I see Eunice Nano is writing that said, good afternoon. This is Elam Tade. I'm using a friend's laptop and can't change her name. Okay, all right. That's okay. We continue with the second factor, which is substrate concentration. So the first one was enzyme concentration. Now we are looking at substrate concentration. And here we see something interesting, that again, the rate of the reaction is on the vertical axis and substrate concentration is on the horizontal axis. And what we see is that there is increase in rate of reaction with increasing substrate concentration up to a point, and then you see there is a plateau of the curve to the point where you are seeing that the rate of the reaction is constant, it's no longer changing. Why that? Let's move to the next to have an understanding of why such a phenomenon. So if you look at the beginning of the reaction where you have, so what you see, the bean-like structure is depicting enzyme molecules. So these are enzymes, and you have substrates, which is these pink spheres. So which means the substrates should bind to the enzyme for the enzyme to catalyze the reaction. In the beginning, you see you have less amount of substrates, which means you have some more enzyme molecules which are free. So you see free enzyme molecules. There are no substrates that are bound to them. And as you continue to increase the amount of substrates, the enzyme molecules which were free begin to get occupied with the extra substrates that you introduce into the reaction system, to the point where you have all the enzyme molecules getting engaged. They are all occupied. They will all be occupied with substrate molecules. So what it means is, from the first point to the second point where you are increasing substrate concentration, there will be increasing rate of reaction because you have more products being formed once you have enzymes that will be binding to these substrates to give you products. But as you continue to increase the amount of substrate in the system, now you see that all the enzyme molecules are occupied and you have substrates that no longer have enzyme molecules that they can bind to. In that case, the rate of the reaction will not change even if you continue to add more substrate in the system, because even when you are adding that amount of substrate more and more, there will not be free enzyme molecules to act on those substrates. And so you have the concentration being constant. So that is what explains what we saw, that at a point, even as we continue to add more substrate in the system, the rate of the reaction becomes constant. It no longer changes. That point we refer to as a saturation point. That is where you have the maximum rate of the reaction. Nothing else can change that, even if you continue to be adding more substrates into that mixture. You will not have any change in the rate of the reaction. All right. Next is temperature. Temperature. So we see, usually we have, if we take, say, the human body, for instance, we say that our physiological temperature is 37 degrees Celsius. So it is at that temperature that you find to be optimal for the activity of enzymes. And remember, enzymes, we said, mostly are proteins. Now, if you continue to increase temperature from that 37 degrees above, you know what it means when you are having increase of temperature above that physiological temperature in 38, 39, 40 degrees. I mean, you start to have denaturing of these proteins, which are enzymes. And so those proteins will no longer be functional. What it means is that the rate of the reaction will then be reduced. So it will be reducing as the temperature increases above that optimal temperature. Because optimal temperature can be different for different enzymes in different organisms. Because you can have some thermophilic bacteria that live in very hot environments, even up to about 100 degrees Celsius. You have some thermophilic bacteria in deep sea. I think when you did some DNA extraction, also you used, or I think is it DNA extraction, or you were doing some PCR, you were using some thermophilic enzyme when you were heating the system, So that enzyme is gotten from a thermophilic bacteria because you needed an enzyme that will withstand such high temperature. Now, again, if you lower the temperature, if the temperature goes below the optimal temperature, you also be having the rate of the reaction being reduced because a lesser temperature than the optimal temperature will be resulting into inactivity of the enzyme. The enzyme, that protein will not get denatured, but it will be less and less inactive. Its activity will be inhibited by the reduction in temperature. I mean, it's one of these, the knowledge from this is what is used in some food preservation, say in refrigeration or freezing, for instance, because at that temperature, you have microorganisms that can act on the food, feeding on the biomolecules, say carbohydrates, proteins, lipids, and releasing toxins into the food, which makes the food unwholesome for human consumption. Once you reduce the temperature, those microorganisms, their enzymatic activity will be reduced. So in that way, the food can be preserved for a longer time. It doesn't mean the food cannot get spots. It can get spots with a matter of time. It's just that the enzymatic reaction that the microorganism is undertaking on that food will be slower. And so it's only that it will take a longer time for the food to get spoiled. So if you keep food in a refrigerator for too long a time, it can still get spoiled. Once there are microorganisms already, in the food before you place it into the refrigerator. And even sometimes there are some food products you buy that there is expiry date on it. And it will still be said that it will still be written on the food that when you open it, consume within this number of hours. That's because when it was packaged, it was sealed away, from air. So sort of there was an anaerobic condition created in the food. But the moment you open it, you have exposed it to oxygen. And many of these enzymatic reactions from these microorganisms occur in aerobic conditions. When you have the substrates being exposed to oxygen, you sort of increase the rate of the reaction, which means that you can no longer keep the food until that date that was written on it as expiry date. So you have to consume it within a certain number of hours. If not, you have multiplication of these microbes, which will then release more toxins into the food and make it unwholesome. Now, we can move on to the fourth one, which is pH level. pH level. Here too looks much like what we saw with temperature, where we have an optimal pH. That's when the pH level is reduced, below that optimal pH, the rate of the reaction is reduced. And similarly, if the pH is increased above the optimal level, it can also result into reduction of the rate of enzymatic reaction. Here you see we have different enzymes that have different optimal pH levels. So every enzyme will be having a particular, pH level that it can operate optimally. So for instance, if you take alpha amylase, say salivary amylase in the mouth, we're having pH just hovering around neutral to slightly basic. And you have salivary amylase being active there, breaking down carbohydrates. But if that amylase gets into the stomach where the conditions are acidic, that enzyme becomes less active, becoming active. But in that same condition, you find other enzymes like pepsin that will be acting on proteins, or maybe renin in babies, for instance, that will be acting on bulk proteins, forming thick curds. So different enzymes will have, depending on the condition in which they act, will be active optimally or less active. Even in some instances, if the pH levels are extremely reduced or extremely increased, those can also result into denaturing of those proteins. If the deviation from the optimal condition in which the enzymes act is so large, you can have some denaturing of these proteins occurring. Okay, now we can dive into mechanisms of action of enzymes, looking at two major models. The first will be the lock and key, which is also known as the Fisher model or the Fisher mechanism. And the second will be induced fit or the Koshland model. So let's take the first one, which is the Fisher model. And this was a model that was proposed by he's a German chemist. Actually, he was a Nobel laureate sometime. I think 1902, 1903, somewhere around that time. Well, he proposed the idea that substrates and enzymes have fitting shapes, like like a lock and key, that you have the enzyme having some specific part of it, like the active site, having some fixed shape, which fits that of the substrate, and the substrate fits into that active site of the enzyme before the enzyme is able to act on it. So that's what is being depicted here in this diagram, that you have a substrate and you have an active site of an enzyme, which is fixed, and you have that substrate that binds to the substrate to give you an enzyme substrate complex. And then you can have the reaction proceeding. So he was the first who, Emil Fischer was the first who brought up this model. That's why sometimes it's being referred to as the Fischer model. A lot of times you will just hear the lock and key, because the concept is just like how you have a key getting into a lock. So what it means is that, say you have let's say this is a substrate, and then you have an enzyme, and the two of them are having shapes such that they can fit just like a lock and key. And so once they fit, they form an enzyme substrate complex. So in that case, you have, say, an enzyme molecule that is having some portions like the active sites that fits to the shape of substrate molecules. And once these substrate molecules get bound onto the enzyme to get an enzyme substrate complex, then a reaction occurs between the enzyme, between the substrates that are bound in that active site of the enzyme, because the enzyme is only catalyzing the reaction between these substrates. And once products are formed, the product is then dissociated from the enzyme. So in the end, you have the product and you have the free enzyme that can bind to more substrate molecules and carry out further reactions. The reaction continues until all the substrate molecules are reacted and converted to their products. Now, the problem with that proposal, as time went on, studying enzyme activity, the problem with the proposal was that if, for instance, we take what you see at the top is a depiction of a non-enzymatic reaction, or call it an uncatalyzed reaction, where you have a substrate that's like a metal stick, and the reaction that should be carried out on it is to break that stick into two. So then it means you can propose that the transition state is a bent state, a bent condition of that substrate. So you have a transition state where the stick is bent. And in the end, you have the products, which will be two broken pieces of the metal. So that's the reaction that is to be catalyzed by an enzyme. Now, if using the Fisher's model, we bring, say, a magnet, so it means that the enzyme is being depicted as a magnet that should fit exactly to the substrate, so that you have an enzyme substrate complex. Now, the problem with this model is that, you see, I don't know whether it was your class that I think yes, in this class, because we did something on the reaction energy profile, where we had reactants moving to the transition state with some activation energy before they form a product. So again, based on that principle of the reaction energy profile, I mentioned to you that, you see, molecules exist at different energy levels. And the fact that a molecule is existing at a certain energy level and called as such, that energy level is a stable state. And so those molecules will not have any reason to be forming a product, to be changing into something else, if they were already stable at at their, the stable state in which they were. That's why there was the need to give them some agitation, the activation energy, which raises their energy level to a transition state before they react and form the product, which will be at a lower energy level, which will be a stable state for the product that is formed. So if you take that into consideration, it means that if you are having an enzyme that is perfectly fit to a substrate, and you have an enzyme substrate complex, then obviously that enzyme substrate complex will be so stable that there will not be any need for a product to be formed out of it. So that enzyme substrate complex is not a transition state. It becomes the product. It just binds. There will not be any need, and so no reaction. There will not be any need for any product to be formed, and so you have the reaction stopping over there. Nothing will happen to that. And so then the proposal of Emil Fischer was modernized to say that actually the substrate does not perfectly fit to the enzyme. So you see the enzyme substrate complex, which is being formed below here. You see the substrate is not perfectly fit, but then you now will have the substrate bending to fit into the enzyme to have the transition state. And once it bends, then it means now it is weaker and can be broken. And so the enzyme will now act on it. And then the substrate is broken and you will have your two pieces of metal, which is your product, and you have the enzyme free to continue the reaction. So there was supposed to be rather the transition state that will sort of fit into the substrate, into the enzyme. But at that transition state, already bonds are going to be weak, and so they can be broken. And once they are broken, you can have new bonds formed, and then you have products that will be formed. So Emil Fischer's model was studied further by an American biochemist, Daniel Koshland, who now proposed what is referred to as the induced fit model. It's actually the induced fit model, which is the current model for enzyme studies. So Koshland built on that idea, the proposal of Fischer, to propose dynamism of the structure of the enzyme, to say that the enzyme is not static. It is actually flexible, so that you have the enzyme. So on the right-hand side, you are seeing an enzyme with some active sites, not really fit to the substrate, but this active site is adaptable. It is flexible, so that it is rather the substrate that induces the fitness of the enzyme to it. And so once the substrate binds onto the substrate, you'll find that the enzyme will then sort of change its form, and then you have the enzyme substrate complex. So this is much more depicted in this slide, such that we see that the substrate, so we have, this is an enzyme with some active sites, and that's the substrate. And once the substrate enters into the enzyme, the active site of the enzyme, you'll find that the enzyme then closes, sort of fits well to the substrate, which means the substrate induces the fitness of itself to the enzyme active center. So it is the substrate that induces the fitness of the enzyme. That's why it's referred to as the induced fit model, also known as the Kochland model. So putting all of that together, the mechanism of action of enzymes based on the induced fit model is such that you have a substrate. So if you take a look at these substrates, I mean, depicted to show that the substrate is having some square portion which should be interacting with the enzyme. And it should be interacting with, I mean, the active site of the enzyme, which is having a round shape, which does not fit into the substrate. But in the presence of the substrate, you have conformational changes. What it means is the substrate induces conformational changes so that you now have that active center of the enzyme now taking the shape of the portion of the substrate which will be binding onto it. In that case, you can have an enzyme substrate complex, the transition state, that will proceed to product formation. And so the product later can be released from the enzyme. You have the free enzyme to continue the reaction. So that was the induced fit model that was developed from, I mean, it's a build up on the idea, the proposal of Fisher. Okay, so we've been talking about active sites. So we take some deeper look at what we mean by an active site. So if we have a protein, so protein, which is an enzyme, is a large molecule. I think I mentioned that it's not that at any part of the big protein at all, there will be a reaction occurring, like enzymatic reaction will be getting catalyzed at any part at all, but you have some specific portions of the enzyme where the reaction occurs. So this is what is being depicted here, that we see the large molecule, I mean the large gray molecule, which is having some area shown as blue. And that blue is the active site. That is where the reaction should be occurring. And we see there's a substrate that enters into that active site. It's like a pocket. It's a protein which is folded, and you have that portion there that the substrate can enter. And even in that active center, you have substrate binding site. And what is a substrate binding site? So again, you have a protein which is made up of amino acids. And that protein is folded. And when it's folded, you have some amino acids, some specific amino acids that come close to each other. And these amino acids, for every enzyme, you find some specific amino acids that act as the binding site. I mean, forming that active center or the active site of the enzyme. And it is on these particular amino acids that you have the binding of the substrate. So the substrate will be interacting with some specific amino acids in the active site. That's what we are referring to as the substrate binding site. Right. Now, when you take, enzymes, so far studies of enzymes have shown some specific amino acids to be mostly found in the active center of enzymes. And these are histidine, cysteine, aspartic acid or aspartate, arginine, glutamate or glutamic acid, and serine. These are the common amino acids that are found in the active sites of enzymes. And they are mostly found to be involved in some acid-base catalysis. If you look at these amino acids, I'll urge you to go and look at. So you take this slide and look, and then you take histidine and look at the structure of histidine. Just pay attention to the side chain: cysteine, aspartic acid, arginine, glutamic acid, serine. Pay attention to the side chains. And you will come to realize that, for instance, aspartic acid and glutamic acid, these are having side chains with carboxyl groups. So that's an extra carboxylic acid group they have there. And those carboxylic acid groups, could be acting as acids. Now, similarly, you have arginine, histidine. Histidine is having a side chain with a special type of ring, what we refer to as the imidazole ring. I think when we go ahead, we may see one of these mechanisms that you will see that there is what we refer to as a proton transfer onto a nitrogen inside the histidine ring of the imidazole ring of histidine. We'll see something like that. Sort of it's accepting hydrogen ions. That is like acting as a base, for instance. It can receive and give out. It will receive it, temporarily and give the hydrogen ions out. We'll look at some mechanism of the sort. And we see serine. If you look at the structure of the side chain of serine, also you see there's an OH, it's a hydroxyl group. And that hydroxyl group also, can have some hydrogen bonding with substrates. It can also even give out the hydrogen from that hydroxyl group as a proton. Cysteine will do same. You remember cysteine is one of the amino acids with sulfur in it. So it's just a CH2SH. So that sulfur hydral group can also be active, giving out hydrogen ions. So you see that a lot of the enzymatic reaction which is occurring in the active center is by some acid-base catalysis or acid-base chemistry. In some instances, we'll see things like nucleophilic attack, electrophilic attack, things like that. You will see those things. And that's also understandable because there's transfer of protons in those active sites. So let's take an example. For instance, we take an enzyme, papain. So papain is an enzyme which is involved in the breakdown of peptide bonds. So it breaks down proteins. And because of that, actually, papain is it's an enzyme which is applied in the meat industry, for instance, in meat tenderizers. And here, what do we see? We see this, the green portion is like the protein of the papain molecule, and it's folded. And so we are looking at the active center of papain, where in that active center, we have 2 amino acids. We have cysteine. So you see, the side chain is SH, CH2SH. That's side chain of cysteine. And we are also looking at histidine. And we see the imidazole ring that I was talking about. Now, another interesting thing you should take a look at is, you see there are some numbers here, 25, 159. What do this mean? So you see how we were talking about the fact that you have a primary structure of proteins. That is the sequence of amino acids in a protein chain. So what it means is that if you have amino acids, you are counting them 1, 2, 3 to have that primary structure, the chain. Cysteine will be at the 25th position, and histidine at 159 position. It is these two. So you see, it is folding of the protein molecule that brings this cysteine 25 and histidine 159 close to each other that way, so that they can be acting as an active center to be interacting with the protein molecule. Now, there's a substrate. It's a protein. I mean, this is just depicting a peptide bond, because here a peptide bond is between a carboxylic acid group and the amine group of another amino acid. So the peptide bond is actually this bond which is between the carbon and that nitrogen. That is to be broken. And what do we see? We see that the sulfur is having some lone pair of electrons that can have, you see what I was saying, can have some nucleophilic attack on this carbon. And at the same time, you are having a hydrogen, which can be having some hydrogen bonding between itself and that nitrogen in the imidazole ring. It's a sort of, creating a condition where there is some sort of a stretch on this bond. The peptide bond, which is between the carbon and the nitrogen, weakening that bond, and in the end, that bond can be broken. The peptide bond can be broken in that instance. I mean, if you look at the mechanism of action into more detail, there will be more steps where you find maybe some hydrolytic reaction will come in. Maybe this hydrogen can get transferred first of all, what I said, a proton transfer, and then later it gives it out, things like that. Step by step, we'll see some of these mechanisms, maybe 2 of them, where you are actually seeing some of these things. All right, so that is an example of active site of an enzyme papain that is acting on a peptide bond and breaking it down. Now, generally, when you take, you're looking at the mechanism of enzymatic reaction, and this is an enzyme, which means you're having, the enzymatic, the active site of the enzyme is the pocket we are seeing there. And you have a substrate. So we have 2 substrates, substrate A, substrate B. These are like the 2 molecules that are to be reacting. They are the reactants. And the enzyme is to catalyze the reaction between these two substrates. Now, let's see the mechanism by which that is done. So starting from left to right, that's a free enzyme and the substrates. Free enzyme and the substrates. Then you will see the substrates are then bonded in the active site of the enzyme. I mean, that's giving you an enzyme substrate complex. And in the active center, you now have the substrates reacting. Substrates are reacting. The enzyme, I mean, if you look back, we talk about the fact that the enzyme reduces the activation energy, which is required for these substrates to react in order for the product to be formed faster. So the substrates react, and when they react, they form products. And the products then get dissociated away from the enzyme. You now will have your free enzyme and the products. And the free enzyme can bind onto more substrates and act on them again, and the reaction continues that way. So if you put all that, what we've been talking about together, this is how the activity of an enzyme, I mean, is carried out. Right. Now we take some specific examples of the mechanism of action of enzymes. We take the mechanism of action of chymotrypsin. Chymotrypsin, that is also an enzyme involved in the metabolism of proteins. And this enzyme acts with a mechanism we refer to as serine protease mechanism. So there are a number of enzymes that act with similar mechanism, what we refer to as the serine protease mechanism. I mean, in the active center, you find serine. That's why it's being referred to as a serine protease mechanism. And you find this with many enzymes which are classified as serine proteases. Now, what you see here, serine. So this is the active center. The active center. We are only taking the active center of an enzyme, which is histidine and serine. Histidine and serine. Those are the two amino acids that are in the active center of the enzyme. And the substrate is a protein. And the substrate too, we are showing it just with, the peptide bond is between an amine group and a carboxylic acid group. So an amine group of one amino acid and the carboxylic acid group of another amino acid. So the peptide bond is what is between the nitrogen and the carbon, which is to be broken. That bond is to be broken. Right. Now, what do we see? We see that there is an interaction between, this hydroxyl group, which is on the side chain of serine, and again, that nitrogen of the imidazole ring of histidine. Now, what you see is that hydrogen from serine, I mean, that hydroxyl group of serine, that hydrogen is transferred onto that nitrogen of the imidazole ring of histidine. So you see there, the hydrogen is now on the nitrogen. And so that nitrogen is now carrying a positive charge. It's a proton transfer. That's what I was referring to, that the activation of some enzymes is by proton transfer, transfer of hydrogen ion. So the transfer is coming from the serine molecule onto the histidine molecule. That activates the enzyme. And once that happens, you now see that hydrogen, which has been transferred onto that nitrogen of histidine, is now having some interaction there. I mean, some intermediate is being formed with the amine group, that nitrogen of the amine group, which is involved in the formation of the peptide bond. And similarly, the carboxylic acid group is also, having some temporary covalent bond there with that oxygen of serine. In the next step, you see that now the bond is broken. The bond is broken between that peptide bond is broken. And you see the side of the protein with that carboxylic acid group, which was involved in the formation of the peptide bond, is still bonded on that serine of the active center. And you see the side of the protein that was having the amine group, the N-terminus in that peptide bond formation is freed. Is freed. Now it's left with this side of the protein with a carboxylic acid group, which is, on this serine residue to be removed. And in that case, you see water has come into the reaction. So there will be some form of a hydrolytic reaction in the end. So the water molecule is having some hydrogen bonding again on that nitrogen of the imidazoles ring of histidine. At the same time, you see also that the hydroxyl group then moves on to that carbon of the carboxylic acid side of the protein which was broken, which is on the serine residue. So you see the OH has come onto that carbon, and the hydrogen is now on that nitrogen of histidine. The reaction continues, and you see what next. Now, from here, you see that there will be the removal of the second side of the protein, which was still on the serine residue with the introduction of water. So you see it's removed. That side is also removed when you have the hydroxyl group from water binding onto that carbon, and it's removed completely. The hydrogen, which was transferred from water onto that nitrogen, now goes back onto the serine molecule. So you now have the enzyme active center coming back to its original active site, regenerated. You see the active site is regenerated. It's gone back to its original form, which means it can go back, take another peptide bond, and the reaction continues. So it continues to break more peptide bonds on and on and on. Now, please don't bother yourself that you want to cramp this thing. Try to understand it. That's all. I mean, the best thing is for you to be able to read chemical reactions. Read the chemical. I mean, it's reading. You read and you can write. So you read a chemical reaction and be able to understand what is ongoing there. So I always say this to students because a lot of times, many of you think that the things that you study, you have to cramp them in your head and be pouring out. But no, you think that I have these things in my head, and when I'm walking about, it's like uh, I cramp it and I want to remember and things. No, I don't have to cramp it in my head. Your head, your brain is important, needed for so many things. So it's not only for chemistry, but understand it. So if you see it, that's like the way you learn to read, so that if you haven't seen any text before and you are giving the text for the first time and you just take it like this, now you start reading it. That's how you are supposed to study, so that when you see something, you'll be able to read it. All right, so that's to say that please don't go thinking that you want to cramp this one in your head, and I will bring something like this in examination that you should do. I don't know what to, I don't know. Maybe I can ask question about, say, which amino acids are found in the active center of chymotrypsin. I mean, that's okay. Or maybe by which type of mechanism does chymotrypsin act in the breakdown of peptide bonds? For you to say it is a serine protease mechanism. I mean, these are simple questions which you can answer. Maybe if you were doing biochemistry major, you are in a university where you are in a biochemistry department, for instance, maybe you may be doing a master's program, for instance. Sometimes maybe the exam can even be oral. There may be a reaction like this can be given to you, and you will explain what is ongoing in that reaction, for instance. But at your level, I mean, you just have to get to know that you have some mechanism as the serine protease mechanism. And what it means is that in the active center, you have the amino acid serine, which is acting as a proton donor. It donates proton onto another amino acid. In this case, it's donating to histidine. So histidine and serine are the amino acids that are in the active center of the enzyme. And the enzyme gets activated by the proton transfer from serine onto histidine. And reaction proceeds step by step with some hydrolytic action along the line. I mean, in the end, it is resulting in a breakdown of a peptide bond in a protein. Okay, we can take one last one. I think this is just enough headache for you today. So we take one last one, which is a mechanism of action of lysozyme. So lysozyme is also one of these enzymes which we classify as glycoside hydrolases. Glycoside hydrolases. You remember when we did classification of enzymes, one of the classes was hydrolases. So the hydrolases were enzymes that catalyze hydrolytic reactions. So hydrolysis. Lysozyme is an antimicrobial enzyme. Naturally, it's produced naturally in our bodies. Like you find lysozyme in the tears that come from our eyes. You find it in, virtually all tissues, because it's needed for our innate immunity against bacterial infection. And so you find lysozyme being, applied in the pharmaceutical industry, in the development of medicines as antimicrobials. You even sometimes, lysozyme is also even used in the food industry for food preservation against bacterial infection. Now, by what action does lysozyme act? So if you take bacteria, a major component of the cell wall of bacteria is peptidoglycan. Now, peptidoglycan is a polysaccharide consisting of N-acetylglucosamine. What you see as NAG is N-acetylglucosamine. And N-acetylmuramic acid, which is NAM. So what you see in that polysaccharide structure is that you have these monosaccharide derivatives which are bonded. So you see N-acetylglucosamine is bonded to N-acetylmuramic acid. And then you see, so they alternate like that in that structure. And you see the bond which is between them is what? A glycosidic bond. So these are some monomers, which are monosaccharide derivatives, which are bonded, having these glycosidic bonds between them. And this structure is a major component of the bacterial cell wall. Now, what lysozyme does is to catalyze the hydrolysis of these glycosidic bond between these monomers. So we see lysozyme there, and water being written to depict that the reaction is a hydrolytic reaction. So we see that this glycosidic bond between N-acetylmuramic acid and N-acetylglucosamine is broken. So by that, what it means is that lysozyme helps to break the glycosidic bond, what is referred to as lysis. So it can also say it's a lytic reaction. They say it's a hydrolytic reaction. So it's breaking down the cell wall of bacteria. So in a sense, it will result into like the death of the bacteria. Bacteria, the cell wall is broken. That is its protection, is sort of a rupture, and then all the components, cytoplasm, everything is out. So the bacteria dies. It's a form of killing, sort of a bactericide, can say that. So the bacteria is damaged, and so it will no longer be there to have its bacterial effects. So that's the action of lysozyme. We see it much more here. So there we see it's okay. There's something being shown here that if you take, say, this is N-acetylglucosamine, for instance. You are having some hydrogen bonding between the N-acetyl group, which is on carbon number 2 there, and you see a hydroxyl group on carbon number 3. There's some hydrogen bonding between these. So hydrogen bonds with this residue that is between the substrate and the enzyme lysozyme. In the active center, so the active site of the enzyme is made up of also 2 amino acids, aspartic acid and glutamic acid. 52 aspartic acid, 52 only means that aspartic acid is on the 52nd position, and glutamic acid is also on the 35th position. So, if those of you who are interested and wants to read more, you can go and look at maybe the active sites of lysozyme, and you see maybe it's aspartic acid, glutamic acid, which will be having some effects. I think I shared a textbook with you. So when you take the slide and you look through, I think you find mechanism of action of lysozyme somewhere, and you may have an information of the sort over there. The bond which is to be broken, which is that glycosidic bond between N-acetylglucosamine and N-acetylmoramic acid, that's what is being referred to as the cleavage site. And that has been magnified for you to see that there is a glycosidic bond between N-acetylmoramic acid and N-acetylglucosamine. Lysozyme comes in that hydrolytic reaction, and you find the two monomers, separated. The bond between them is broken. That way, you have the cell wall of the bacteria broken. That will result into the death of the bacteria. All right, I think I've given you enough headache. I'll end it here. If you have anything to say, no one has written in the chat except for Eunice Nano from the beginning. If you have anything, if there's anything you want me to go over, you can raise your hand. I will enable your mic, and then you can unmute and speak. The floor is open.